Hello and welcome to this recording titled Introduction to Exposure. The objectives of this presentation are for you to understand the objectives of characteristics and uses of exposure information. Right. There's some good reading available on exposure assessment. This document from EPA in 2019 is a nice introduction to many concepts in exposure assessment, so I recommend looking through it. Let's start with, it's necessary to recognize that we live amidst numerous types of hazards to our health. Some of them are chemical, others are biological, and even others are physical hazards. In the field of environmental health involves understanding how our living conditions influence our health. And these conditions include the 160 million chemicals known to humans, the 1 trillion species of microorganisms thought to inhabit Earth, and myriad physical phenomena like the various energy forms that make up the electromagnetic spectrum. So environmental health regularly deals with the quantity of these materials that exist inside people, around people, and are related to things that people do. These considerations all address exposure to these stressors. And exposure is the subject of this introduction. So, what's the definition of exposure? And classically, in environmental health, exposure is defined as contact between a stressor and a person. And that contact can take place at an exposure surface, like the mouth, the airways, our skin, our eyes, and it can take place over a certain period of time, which we can refer to as the exposure period. And that contact at the surface of our body or surfaces of our body doesn't just happen willy-nilly, right? Of course, it happens as we come into contact with air we breathe, water we drink, food we eat, soil that might be on our skin, or other materials like consumer products that contain a stressor. So that's a definition of exposure. How about exposure assessment? That phrase refers to the process of estimating or measuring the magnitude, duration, and frequency of exposure to a stressor, along with the number of people exposed and the characteristics of those people, like where they are, what they do, what other risk-related features they may have, like pre-existing disease, for example. Exposure assessment also describes the sources, pathways, routes of travel from that a contaminant takes, and any uncertainties in the assessment. Human exposure falls right in the center of this environmental health paradigm that by now I think you're probably familiar with, right? It's exposure lies between concentrations of substances in the environment and internal doses or internal amounts of those same substances or their byproducts in the body. Right? So human exposure is a critical piece of understanding human health risks. Exposure has 4 key dimensions. They are shown here on this slide: magnitude, duration, frequency, and really timing or life stage. Magnitude typically refers to the amount of some hazardous substance in an environmental medium like air or some consumer product. Right? And that amount we typically characterize as a concentration. So the amount of that substance per the volume of the medium, or the mass of the medium, or another measure of the size of that medium, and we'll look at that a little more closely in a second. Duration just refers to the period of time over which an individual or a population may be exposed. Sometimes we're interested in exposures over a lifetime. Other times we're interested in exposures only over a discrete period of time. And that time period that we're interested in primarily relates to the toxicological properties of the stressor. Like, is it a carcinogen or a suspected carcinogen or not? Frequency of exposure is the rate at which exposure events occur over the duration of exposure. The frequency could be all the time, right? In that case, frequency happens continuously. Or that frequency could be intermittent, say once a week, once a month, once a year, right, etc. And life stage, or timing, refers to when in the life history of an organism this contact or exposure occurs. Why do we care about when? Well, the susceptibility of humans and other organisms to toxicants is somewhat dependent on where in the life stage that exposure occurs. Generally speaking, periods of conception and gestation and early development of key organ systems like the central nervous system are periods of greater susceptibility to stressors. Similarly, elderly folks can be, not always, but can be more susceptible, and certainly people who have a pre-existing disease are more susceptible in many cases. Another important part of exposure to be aware of is this concept of exposure pathways. So an exposure pathway is the course, meaning the route, that an agent follows from the where it was released to the environment all the way through to the so-called receptor, right? And here in public health and environmental health, we're usually interested in human receptors. So it's the pathway that this substance takes. Some pathways are shown on this diagram. Like, for example, we have some over here on the left where my cursor is. There's some drums of maybe some volatile substances that contain volatile substances, and those volatilize to the air, and they get carried by air currents in the atmosphere, and they may make their way to where people are. And subsequently there could be inhalation and actual exposure occurring, right? So that would be one pathway. These drums might also contain substances that are not so volatile and have some affinity for water, and they permeate into the soil, and they may travel through the soil, gas actually, right? So the space between grains of soil. And if those grains aren't filled with water, then those pores aren't filled with water, they tend to be filled with air. And so volatile compounds can actually move through those voids in soil. Or they may, and eventually that soil gas could be taken up into, say, like the basement or lower level of a building, after which people could then be exposed. So there's another pathway, and there's a groundwater pathway shown on this diagram too. So exposure pathways are super important for a couple reasons. One, it's vital to determine whether an exposure pathway is complete or incomplete, because an incomplete pathway means contamination in the environment, environmental concentrations, right, of this stressor, but not necessarily human contact. A complete exposure pathway is one where human contact occurs. That's important because without a complete pathway, no exposure occurs, and that means no risk exists. Right? Second reason that understanding an exposure pathway is important, because if you know how the substance moves from a source to the people, you then have the information needed to help you begin thinking about ways to intervene, right? Where in that pathway is it possible to reduce transport, or to reduce emissions, or to reduce the actual exposure, right? By understanding the pathway, you can figure out where to intervene. Now, the end of a complete pathway of exposure is typically an exposure concentration, meaning the amount of that substance in air, water, food, etc. So let's take a quick look at concentration. And here we're just going to briefly examine the types of units that one encounters for expressing concentrations of stressors in environmental health. So this table has a lot of cells, obviously, and there's a lot of information in here. And so starting in the left column, we have some common analytes like organic and inorganic chemicals and elements and things like that. And in the row below that, we have other types of stressors, like ones that aren't really measured in terms of mass, but more measured by quantity, like they're countable. These include things like fibers, like asbestos fibers, or spores, like fungal spores or bacteria spores, or even viable bacteria and fungi. Viable means that the fungi and bacteria can reproduce, right? They can grow. And then the third category here is radionuclides. And the variety of different common units or metrics used to describe concentrations of this first group of organic and inorganic compounds. We have some concentrations expressed as weight per volume, like milligram per cubic meter of air, or milligram per liter of water, right? We have some other metrics that are actually ratios, like a volume to volume ratio. Those apply to concentrations in air, and they're typically expressed with terms like parts per million and parts per billion. We're going to talk about those in detail in a separate recording, but I just want you to be aware of the different types of concentration units at this point. Another metric is weight per weight, which is akin to volume per volume, but obviously based on weight. And these mixing ratios are generally used for contaminants in water and food and soil. All right. Sometimes in environmental health, an exposure measure is actually the weight per unit area, like micrograms of lead per square centimeter of floor material in a house. Right? That would be called a loading. And then these discrete or accountable objects, they're generally more, the units are more like the number of countable items per volume of air or per volume of water, and sometimes it's per area. And then radionuclides, they have their own language of units that have to do with radioactivity. That's what Curie is and a becquerel. And I'll leave that for later in the semester. So at this point, we've talked about concentrations, and we've talked about pathways. And now we're at the point where the contact might actually occur. And there are three routes of exposure for people. Dermal, so contact with the skin. Ingestion, meaning intake through the GI tract, right, through the mouth. And inhalation, right? Those are the three routes of exposure. dermal ingestion, inhalation. So if you have a substance in the air and people are present where that substance also exists in the air, we have the possibility of exposure as long as the people are breathing. Well, of course they're breathing. But what is the rate of exposure? How much intake of that substance in the air occurs per unit time, like per minute, per hour, per day, per lifetime, right? How much actual intake occurs? Well, to go from that environmental exposure concentration to intake via one of these three routes, we need to know something about the amount of air people breathe, the amount of water they drink, the amount of food they eat, and so on, right? That area of research in environmental health is termed exposure factors, okay? These would be factors that influence or modify exposure. So some of the key and core exposure factors are shown on this slide, like inhalation rates for people, drinking water consumption rates, body weights, food consumption, types and amounts. So here are some values for these exposure factors that are recommended by international organizations like the World Health Organization and the International Program on Chemical Safety, which resides within WHO. Those organizations recommend an inhalation rate of 22 cubic meters of air per day for an adult person. So that's just about a cubic meter per hour. They recommend a drinking water consumption rate of 2 liters per day. IPCS says more like 1.4 liters per day. They recommend body weights of around 60 kilograms. And you can find online from WHO diets, including types of food and amount of food eaten for geographic clusters of countries. Now, these are not there are other sources of exposure factors. In fact, the U.S. Environmental Protection Agency has an extensive program on characterizing exposure factors for people in the United States. China, the Environmental Protection Agency there, has its own detailed characterization of exposure factors for people in that country. So this is the last slide in this introduction, this kind of overview of exposure assessment. And the goal of this slide is to introduce you to this notion of exposure-based environmental health benchmarks. What does that mean? It means the following. If we have an understanding through science and through science policy of acceptable levels of exposure to a substance, meaning like a level that we think is unlikely to result in adverse health effects, then we can express that amount of acceptable exposure, kind of target exposure, in units like concentration. That's an exposure unit, right? Or we can express it in units of intake, like milligrams of some stressor per day. Like the objective would be to keep the intake rates below this health protective guideline value. So there are many forms of these exposure-based health protective guideline values, and this slide is a representative collection of these exposure-based environmental health benchmarks. So we'll look at these closer. We've already looked at some of these in terms like the cancer slope factor and the reference dose in the context of dose response analysis, and we'll look at them more closely as we move ahead. Okay. All right, so the summary of this introduction is the following. Health risk is a function of exposure to a stressor and the toxicity of that stressor. Exposure requires the joint presence of a stressor and people. Exposure is typically quantified by the magnitude, like the concentration, frequency, and duration of human contact with a stressor. Exposure results from transport of a stressor via one or more pathways. When people have simultaneous exposure to a single stressor through more than one pathway, we can add those up, and we refer to that sum as aggregate exposure. The routes of human exposure are inhalation, ingestion, and skin contact, so a complete pathway of exposure culminates with intake through one or more of these routes. And if we have a decent understanding, right, a reasonable understanding, a science-based understanding of the toxicity of substances in our environment, we can actually express the health protective levels of those same stressors in units or in terms of exposure. All right, that's it. See you next time.